Welcome to the lab. I'm a PhD student at the Technical University of Munich. My name is Lukas Möbus. I will show you today the lab where I study the effects of breathing on our autonomic nervous system, so how we can self-regulate autonomic arousal with breath control. So when you hear Technical University of Munich, you might have the expectation of a very big lab with MRI machines and the Alps in the background. But here we don't need all of this fancy equipment to actually do the measurements that we need in order to answer the research questions that we have. So we have a very simple setup here with the Nexus 10 MK2, a second monitor, and our computer that does all the work. So every scientific endeavor starts with a research question. You have a burning question in mind that you try to get the answer to. But in order to get an answer to your question, you need to formulate hypotheses and then also operationalize the constructs that you try to measure. So if you're interested in relaxation, you need methods to make relaxation measurable. So you have the biological processes that are going on in the body invisibly, and you make them visibly with the methods you use. But what is relaxation and how can I measure relaxation? We can measure relaxation on multiple levels. On the level of the central nervous system, the autonomic nervous system, and also self-report. What people report, how relaxed they feel. And we try to measure relaxation in all those levels. We collect EEG data to measure neurophysiological activity, so that is the electrical activity that goes on in our brain all the time, even when we are asleep. We are collecting data on a physiological level. For example, how sweaty are your hands, or how is your heart rate variability, which are indicators of your autonomic nervous system, the activity of your parasympathetic and sympathetic nervous system. And we ask people with a standardized questionnaire, how relaxed do they feel? For example, do their muscles feel tense? Do they feel relaxed and calm? Etc. So you have your research question, you have your hypotheses, you also have the methods to measure the construct of relaxation. And now I'm going to take you through the whole process of how you can actually answer your research question by using the methods that we have here in our lab. So first, you welcome your participants into the laboratory, and then most importantly, they will sign an informed consent, where they are fully informed about what is going to happen to them when they are participating in a scientific study, so that they are consenting, fully informed on what is about to happen in the scientific study. So welcome to today's experiment. In front of you, you have the informed consent sheet that provides you with all the information about the study, and where you will sign at the end that you're consenting to being part of the scientific study. After they sign the informed consent form, I will set up all the equipment. This takes a little while because we are collecting neurophysiological, physiological, and self-report data. So the first sensor that we attached is the chest strap. It measures the breathing frequency of the participant, and it should be tight but not too tight so it interferes with the breathing. And now I will take the non-dominant hand of the participant and place it on the mat, where I will now attach the various sensors that we talked about, like the temperature sensor, for example. I will place this on the small finger. To measure peripheral temperature. Then I will take the galvanic skin response sensors and place them on the index finger and the ring finger. And lastly, I will take the blood volume pulse sensor to measure heart rate variability and place it on the middle finger. Now I make sure that the cables are out of the way A little bit of mess is unavoidable, but we keep it manageable. And this is the setup to measure the activity of the autonomic nervous system. First, I will measure your head's circumference. For that, I will place a measuring tape around the head. And now with that measurement, I can select the fitting cap. Now that I have this electrode cap that holds the electrodes in place, I put this over her head. And I always asked, how does it feel? Does it cut into your skin in any way or feels uncomfortable?
Feels good.
And before I clean the places where I'm about to place the electrodes, I will double check if the cap is fitting correctly. So I take the distance from the inion to the nasion, and this fits almost perfectly. I'll just push it a little bit back. And now I also check whether it's correct laterally. That's also almost perfect. It's 0.5 centimeter to the right. So now that the cap is fitted, I will clean the places where I will put the electrodes to measure the electrical activity of her brain. For that, I need the new prep. We will place reference electrodes on the earlobe. So I begin by cleaning the earlobe of dust or old particles of skin. Also here on the left side, I will place the ground reference electrode on a bone right here at the back, os mastoideum. Okay, now that I cleaned the places where I am about to place the electrodes, I will get the electrodes and place them first over her shoulder so I get some order into the whole setup. Now I take this conductive paste and put it on the electrodes. That improves the conductivity from the skull and the scalp to the electrode, so we can have a good signal of the electrical activity of the brain. I try to parse the hairs apart a little bit so I get as close to the scalp as possible to get a good signal. And lastly, we have the ground reference electrode. So now that we set up the electroencephalography, we can put the equipment away that we needed to set everything up. And begin with the experiment. We'll start with a resting measure, when we just want to see what is the baseline activity in the central nervous system, in the autonomic nervous system, and what they report on how relaxed they feel or how stressed they feel. So now we are opening the BioTrace software. We select the screen and we begin our recording by selecting our participant with her pseudonym. We start our recording, and as you can see, the various sensors right away collect neurophysiological and physiological data. Here you can see the active electrodes placed on the prefrontal cortex. You can also see muscular activity. In this case it's an eye blink. You see the blood volume pulse, so this is the heart rate. And we see the respiration activity, so the inhalation and exhalation. We have a respiration rate of about 18 right now, and the heart rate around 70 to 90, which is also displayed here as a graph. And down at the bottom we can see the skin conductance and temperature, which is at 0.64 microsiemens and 25.69 Celsius degree. Before we begin the baseline measurement or resting measurement, I will select the screen. In this case, it's the baseline screen, and I will open the screen in the secondary monitor. And now that the screen is open, I will press enter and mark this as the resting measurement. Now, after 5 minutes, we set another marker for the end of the resting measure. Now we have a baseline of the activity of the central nervous system and the autonomic nervous system. And we also want to get a baseline of her sense of relaxation right now, so the subjective sense of relaxation. That's why I'm now dragging this questionnaire over to the second monitor, where she will be filling out this questionnaire. Now that we have the baseline measurement of the central nervous system, the autonomic nervous system, and the self-reported feelings of relaxation, we can begin with our intervention, because our intervention is there to answer our research question. In my case, I want to know what are the effects of a breathing exercise on the central nervous system, the autonomic nervous system, and the self-reported state of relaxation and stress. Now, from the baseline measurements, I calculate the spontaneous breathing frequency I'll change the screen to look at the data that we collected so far. I select the segment during the baseline measurement. I compute the statistics for this segment and I take the mean of the breathing frequency throughout the baseline measurement, which is 19.22. And I put this value into Excel, where I now see the breathing frequency for our breathing exercise displayed down here. So that would be 2.6 seconds inhalation and 5.2 seconds exhalation. These numbers I need in order to set up the pacer in BioTrace again. Now back in BioTrace, I change back into my monitoring screen, and now I load the screen for the breathing protocol. Again, here I open it in the second screen. Here we have a pacer that displays the breathing frequency as well as the breathing rhythm. The breathing frequency will be reduced to 40% of her spontaneous breathing rate. So it's already personalized to her individual breathing rate. The exhalation will be twice as long as the inhalation. And I configure the pacer settings. So now, based on her personal spontaneous breathing frequency, I set up the pacer for her individualized breathing protocol. And now that everything is set up, I press OK and the breathing exercise starts now. So we will see today whether breathing exercises have a measurable effect on relaxation. So do we see changes in the central nervous system's activity, in the autonomic nervous system's activity, and the self-reported state of relaxation? So we have multiple measurements that we take in order to see whether the participant actually is more relaxed after the breathing exercises than during the baseline measurement. We have peripheral temperature, which we measure with a temperature sensor. We would expect that if participants are more relaxed after the breathing exercise, that temperature in the periphery rises, because the sympathetic nervous system, so the part of the autonomic nervous system that is responsible for the flight, fight, or freeze response, or generally the part of the autonomic nervous system that gets us activated, actually inhibits blood flow to the periphery, like the fingers. And when we're really relaxed and the sympathetic tone releases or decreases, then we have more blood flow into the periphery. We also measure skin conductance, another measurement of sympathetic tone. So we would expect, if participants are really relaxed, that their skin conductance decreases due to the relaxation. And we would also expect an increase in heart rate variability, which is a measure of parasympathetic activity to the heart, which we measure with the blood volume pulse, so changes in light due to the diastolic and systolic activity of the heart. So with the 4 electrodes placed on the prefrontal cortex that measure the electrical activity of the brain, we do a frequency band analysis where we look at beta, alpha, and theta activity in the brain, which are indicators of central nervous system arousal. So we would expect that there's an increase in alpha and theta activity when participants are more relaxed, for example, after the breathing exercise. Now, some of you also think, hey, why would you bring on all these different kind of sensors on the participants' head and on their hands? Why not just ask them how they feel after the breathing exercise? And we do that too. So we have standardized questionnaires like the relaxation state questionnaire that measures relaxation via self-report. So this is a questionnaire that has multiple items where the participant is asked about their state of relaxation. For example, how tense their muscles are, or how calm and relaxed they feel right now. And this gives us an idea of how relaxed the participant feels. Another important parameter is breathing deep into the abdomen, because this has also been shown to have relaxing effects. So breathing up into the chest might have stress-inducing effects, whereas breathing deep into the abdomen might have calming effects, or the literature has shown it to have more calming effects. So I will take you step by step through the process of how we are going to collect the data, analyze the data, and interpret the data. Again, similar to the baseline measurement, I set a marker to denote the start of the breathing protocol. And as you can see here, the respiration rate already adapted to the breathing protocol. It is now at 7.6, so it's a lot slower than the spontaneous breathing rate of 19.22. And you can also see a phenomena that is referred to as the resonance frequency, where the fluctuation in heart rate and the fluctuation in the breathing rate somewhat resonate, so they show a similar pattern of increase and decrease. This is also referred to as the respiratory sinus arrhythmia. Now, after 5 minutes, we end the breathing protocol and set a marker. So right after the breathing exercise is finished, I will open up the questionnaire again, and she can continue to fill out the questionnaire. It is the same questionnaire she filled out at baseline. First, we make ourselves an impression of the data we collected. Obviously, we need to take into account that the context in which we collected the data was quite artificial. So we had the stress induction of having a camera team around and the participant doing the breathing exercises in this context. So first, we take a look at the resting measurement. We can zoom into that segment and have a closer look at how this data looks. So there's actually a reduction in the skin conductance just by staring at the fixation cross and basically having no instructions whatsoever. So this can already be relaxing for some people. We also see that the breathing is quite rhythmic and that there's not as much fluctuation in the breathing rate as during other periods before and after the resting measurement. In order to get a sense of the EEG data, we would have to do further analysis. But before we do that, let's have a look at the heart rate variability during this period. So I select the segment again, and we can do some easy statistics here at the beginning by just looking at the mean heart rate variability during the baseline measurement, which was 25 milliseconds. And then we can compare this with our breathing exercise. So you can keep in mind 25 milliseconds, and let's have a look at our breathing exercise. Now back at the overview, we can take a look at the breathing exercise and the physiological and neurophysiological data we collected here. We again zoom into our data and have a first impression of how it looks. So the skin conductance, as you can see here in yellow, does show a reduction, which means the sympathetic tone decreased, which is indicative of a relaxation response, although the temperature does not behave as expectedly or does not show values that we would expect. It actually decreases, which might be an indicator of stress or in this case just a normal fluctuation in temperature, which could also be the case. Now down at the bottom we see high compliance with the breathing exercise, so the breathing frequency does not change much throughout the breathing protocol because it is instructed by a pacer. You see this little glitch here where we did some setup changes during the filming. You can also see that the breathing, the inhalation and the exhalation, as well as the increase and decrease in heart rate are fairly aligned here. So this is what we refer to as the resonance frequency. And my guess would be that heart rate variability is increased here. So in the baseline measurement, we had a heart rate variability of 25. Now here we have a heart rate variability of 41. So heart rate variability almost doubled during the breathing exercise. And that is indicative of a relaxation response, so increased parasympathetic activity in the autonomic nervous system. And again, by just looking at the data, you cannot really make sense of the EEG data. I could show you a sneak peek into how it looks when we look into the data more closely. So here we see at the top the EEG activity, and we even have to zoom in further to really be able to make some sense of what is going on in this data. So as prefaced, we can't really make sense of the EEG data by just looking at it. Just seeing the frequency and amplitude doesn't tell us much. That's why we will do a fast Fourier transformation. So we will select this segment and we use the analysis function and do a fast Fourier transformation. The setup is already as we want it to be, so we can export right away. We can label our data. Open it with Excel, and now you have this Excel sheet where you can pick out the measure that is most interesting to you. So we are looking at relative changes in absolute power because that gives us a good idea of how the activity of the brain changes within frequency bands relative to the frequency bands. So whether we have an increase in theta, beta or alpha, and we can here select our values that we are interested in, and then we would compare them between the two breathing exercises to see if we have a difference in activity in the brain during a baseline measurement and during a breathing exercise to induce relaxation. We would compare whether the relative power in the alpha, theta and beta frequency changes between the resting measurement and the period during the breathing exercise, and we would expect that the relative power of alpha and theta increases during the breathing exercise, which would be indicative of less arousal in the central nervous system, in this case in the prefrontal cortex, which would go along with a relaxation response. And we would expect that the beta frequency, which is more related to arousal, to emotional processing, also to being very alert, that this would decrease during the breathing exercise, which would be indicative of a relaxation response. So we got a first impression of the data that we collected. We had a look at the resting measurement. We had a look at the data during the breathing exercise. It is very important, however, to mention some of the caveats when it comes to interpreting the data that we just collected. For example, we have just the context, the setup here. We are filming this data collection, so there are some external pressures put on the participant as well as the researcher, which might change the data collection, as well as how the participant feels. So it might take more to feel comfortable. It might be more difficult to truly induce a relaxation response due to the circumstances. And we only have a single participant, so there is large variability in the response to any intervention that humans take part in. So you would have to take into account just inter-individual variance in response to any behavioral or pharmacological intervention. Additionally, one caveat could also be the experience a person has with relaxation techniques. So people that are experienced with relaxation techniques, such as breathing exercises, usually do show a stronger response to such an exercise because the body is already familiar with the intervention and the neuronal pathways to induce the relaxation response are already stronger, well formed, and you would see a stronger response in participants with experience, which is also very important because it ties everything together in a scientific study. You not only want to measure the things that you are interested in, like relaxation, but also confounding effects. Like we would measure that there is a setting in which we do this experiment. How much experience do the participants have with relaxation techniques? And how large does my sample size needs to be in order to detect the effect that I'm expecting? So these are all interesting questions and caveats you have to take into consideration when you're conducting a scientific study. Collecting data, interpreting your results, looking at confounding effects, that's all very important. But what might be even more important, or what is more important to me, is what is the effect the research I'm conducting has on other people. So in this case, we try to develop personalized breathing exercises that help people self-regulate their autonomic arousal in order to cope better with stress. Because chronic stress is oftentimes the antecedent of chronic illnesses, such as depression, anxiety, but also metabolic issues, such as obesity or other factors. Because stress really enhances the progression of disease, as well as pushes people over the edge to develop a disease. So you're exposed to chronic stress for a long period of time, and then you might get a panic attack. In order to prevent that, we need strategies like breathing exercises that are tailored to our needs and our physiology that help us to cope with the stress that we experience in our lives.